So, all right, start again, restart. So thank you, Marinka, for inviting me. Thank you, Ray and Marinka, for organizing this. This is a really good idea. I know it's a lot of work to organize this and make it free for everybody. So as a participant, again, thank you. You guys did a wonderful job doing this, so thanks. I don't know my audience, so I prepared this for both academic and industry colleagues of mine. Hopefully, I can serve both of them reasonably well. As you can see, the title of my talk is Acceleration of Drug Development. By drug development, I mean, very generally, it goes from early drug discovery to all the way to marketing a drug. So that entire process. Typically, that's not really called as drug development, but the whole process is what I'm calling drug development here. And by AI, if you're not an expert in AI, just imagine AI as machine learning, and that's fine. If you are an expert and you're like me, who cringe in using AI as a word, I would use the definition that anything that is either supervised learning or reinforcement learning that doesn't require feature engineering is what I'm going to call AI in this talk. And everybody knows data science. We can have a long conversation on that, so you get the idea. So with no further ado, I'll just get on with it. The drug discovery and development, if you're not in the industry, you already know still that it's a long process. It takes a lot of money. There's a calculation that's costs up to $2.6 billion per average for each drug. And there's a lot of things that goes on in that process. Okay, so if you look at the early phase of the research, which is where most people are, particularly if you're a small biotech or academia, you're playing in the early phase of research. We're trying to identify a drug, not really a drug, a compound that actually can bind to the target of interest. And in a company like AstraZeneca, that means that you're going through lots of different compounds, 5,000, 10,000 compounds, before it actually gets to the stage of going into development in clinic. So 4 to 6 years in the early development, then many more years in the clinical development, right? And during this phase, a lot of drugs simply fail, and for many different reasons. And that's something that I'll go to later. Here is something that a lot of people really miss. The idea about why drugs fail or succeed. So efficacy and safety are extremely two important things for a drug to either make or break. Efficacy is obviously seen as something, the drug actually works. What does it mean by the drug actually works, right? You need to have the right target. So let me just check 1 second. Windows Pointer. This one. All right. So you need to have the right target, just like Robert was talking about. And many times we get the target wrong. And a lot of the times most people forget is really the safety. AstraZeneca did a big exercise in 2014, trying to understand what happened to their drugs. And one of the things that came from that article was that in their analysis, most of the drugs fail for one of these reasons at a 1:1 ratio approximate, which is that either safety or efficacy. And safety can be because this drug has off-target effects, or it causes things like liver failure, kidney failure, et cetera, et cetera. So what I'm really after is, can we as a community use AI to make drug development better, faster, and cheaper? So you probably think about what are the areas that AI can really help, and that's something I promised in the talk that I'll deliver. And I'll mix and match during this talk where the opportunities are. So there are 5 key pillars that I see where AI can help. One is, as Robert was talking about, there's a target identification. The second one is something that almost every academic is playing in the AI space for pharma, is drug design. The third piece is toxicology. Fourth piece is imaging. Imaging is not really part of a process in pharma. There's a lot of places where imaging comes in. But I put imaging in between toxicology and clinical because imaging plays a huge role in toxicology space as well as in clinical space. And there's a lot we can do in that imaging space. And then finally, the clinical. I'm not going to be able to talk about everything in this talk, and I'm going to skip the target identification piece of this and also the clinical piece of it. So I'm going to primarily focus on drug design, toxicology, and imaging for this talk. So here comes some use cases. Let's look at the idea of drug design. Can AI accelerate drug design phase in pharma? And I know many of you are aware of a lot of papers in this field, but how does it actually work in a true pharma like AstraZeneca? And before I go on, I want to warn you, is that drug discovery is a very complex multi-parameter optimization problem. There's a lot that goes into developing a drug, and even when we say drug discovery in pharma. It's not just identifying that first compound that binds to your target and then you call it a drug. It's not a drug. There's many things need to happen. We'll touch base on that. So there's a thing called as DMTA. DMTA is, in pharma, it's known as design, make, test, analyze cycle. So what happens in DMTA? You have a drug target you want to target. And as I said before, you may not necessarily know that the target is actually correct. A target is only truly validated in clinic. You have identified maybe a compound that is weakly active against that drug targets. You may be in the micromolar range, it may be worse than that, millimolar range, whatever, right? But you have something that binds that target, but it's not a drug. And so the first thing to do is look at the drug, what do we know about it? We may know certain things about the toxicity risk of that based on some competitive intelligence or the class of the compounds, et cetera, et cetera. And we take that and you go into the design phase of making the molecule or designing how to make the molecule, test that molecule. Testing means simply at that point, testing very limited number of parameters. This could be the affinity primarily at that point. And then you analyze the data and then you design again, you keep doing this iteration until you get a candidate drug that is highly potent. It's effective, at least in vivo models. It's metabolically stable. You don't want explosives or things that you need to store at a very low temperature, for instance. No toxicity issues, at least at that point. But many things need to go into the toxicity piece. We'll come to that later. And then this process takes about 3 years. At AZ, we have really embraced AI as a piece. And since 2019, we have implemented some pipelines that allows us to improve our drug discovery and development process. So from the drug design process, if you think about most companies, they all have something along these 4 lines, or the first 3, which is virtual screening, and STS, which is high throughput screening, FPLG, which is fragment-based design, and then you have other modalities that could be a DNA encoder library, et cetera, et cetera. These are all things that AZ always used, nothing new here. We also use computational biology methods, computational chemistry method for a long time. So there's nothing really new in that AI's piece of thing until we really implemented deep learning based algorithms to enable our new molecule generation. This is truly beginning to change how we invent things. And I personally believe this will fundamentally transform the industry. And this is a major contribution that all the computer scientists in the deep learning space can be very happy about, that this is a real true contribution that has occurred to pharma. So I'll talk about this in a minute, but we have published a bunch of papers in this space. Again, this work is not mine. There's a whole bunch of people have worked on this. There are some publications that I put here. If you're interested, you can go and read. there are different pieces of this that we have developed in terms of the deep learning space. So what is the problem here? When you think about it, you might think, oh, well, deep learning has really been very powerful. Why don't you just apply, make some molecules, and then go test it? Well, it doesn't really work that way. Here's an example. Just 3 years ago, I am not going to name the paper or anything, a leading, a very influential piece of work came through. And the first version of the paper had these structures in it. If you look at the structures, and this is based on machine learning and deep learning, when you look at this, they look nice if you're a non-chemist. But I'll tell you, if you're a chemist, if you synthesize some of these molecules, you probably win a Nobel Prize, and most of it won't make a drug ever because they will be explosives. So computer make this drug-like molecules that are not really ever going to make a drug. So that's the problem that AZ needed to solve. And so how did we approach this problem? So the idea is to borrow information from things like natural language processing, where RNNs, or recurrent neural networks, were being utilized, and where you might type something like the grass is, and you know that the next word most likely is going to be green, or the grass is growing, etc. If you're talking to an adult, you probably will be focused on grass is growing versus grass is green. Or if you're talking to a kid or teaching kids, you probably would be focused on grass is green. So there's a context dependency here too. And that brings me to the transfer learning piece of it, which briefly I touched base on. So the idea is you have conditional probability that if, given the fact that the first 3 letters with the words with the grass and is, what is the probability that it would be green, or the probability of green given is grass in the previous sentences, sort of like a Markov model if you know that. So the same thing can be actually utilized here, is where you have molecules, you have a C following a C with a double bond. What would the next molecule should be? And that's basically, okay, if it's an O, what would the probability that it's an O given a double bond and a C and a C? So that's the idea that we use, is you look at real molecules and derive this probability distributions and use the probability distribution to make molecules rather than just simply make molecules. And how do we do that? And for the non-experts in the space, we can convert a 2-dimensional molecule like this into a small space, which is basically a string. It exactly looks like a string. You have C, O, etc., 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 and follows. So it's like language. You can apply RNN to this language, and you can learn this language. So here's how it sort of works. The idea of generating a molecule starts with these vocabulary that you put on the y-axis, and you have probability distribution that knows that, okay, what is the prior probability for each of this, and if I start now with the next molecule, next atom, you will be sampling from this probability space, and you might sample, okay, I'll take the sample, this is not the highest, but I'm sampling, so I'll take oxygen as the next, and I might pick carbon as the first and oxygen as the next, and then I continue building this. Then once I have oxygen as the next, I have another probability distribution. What comes after that? One of these are this probability distribution for the oxygen. So then I sample that. So I continue sampling this, and I start to build this whole thing, and you start to see this molecule being built. If you missed it, I'll just play again. It's like this. So this is all open source. The publication I put in the bottom. You can read more about it. So that's the general idea about it. That's how you create the model. But it's really not the end of it. There are many other things that goes into it. I won't have the time to go through all the details, but you get the general idea. There need to be additional filtering, etc. But I do want to show an example, a true example of where we actually made this really work within AZ. So here's a project, and we have many projects that we're working on. One specific target, we want to make a molecule against this target. So we first generates 800,000 compounds. These are all virtual compounds, and then we need to filter that. So this 800,000 compounds are split into 3 buckets: one with some reinforcement learning, one for fixing the scaffold, if you know the chemistry of it, not important, and then the other was transfer learning, really focusing that learning on specific set of molecules. So with that 800,000 molecules and going through this, starting with this, we have we can filter out to really identify representatives, because you have a lot of redundancy in it, and so how close the molecules are, etc. So you create a similarity filter limited to over 368 compounds, and then you do some additional filtration. You know, what could be the activity, etc., etc. What's the docking score, etc. You come out about 58,000 compounds, but that's still too many compounds. This is where the real challenge is, right? AI is suggesting a lot of compounds for us. There's no way to make these compounds. So at this point, you cluster these compounds, identify cluster spaces, and take representatives from this cluster that actually makes sense to actually test. And that's a combination of human power, mental power, the chemistry power, etc., right? Because it's not just a simple AI, go make these compounds, because sometimes, as I said, some of those can get explosives or they're simply not synthesizable. So all of this goes into the equation of how we select it. So from that point, it requires some amount of effort. So the point is, the team embraced this. They took the AI-based compounds, and then they started testing it. So the first iteration is in the first 2 columns here. And as I said, it's a multi-parameter optimization. We look at different characteristics of the compound as we move through the pipeline. In the early phase, you're not looking at too many characteristics. You're looking at, you know, IC50, log D, clearance, etc., etc. So first iteration is the first 2 columns. The green is good, orange is not so, red is definitely bad. You don't want to be in that range. First iteration, after the 1.1 iteration, it was already reasonably good, only had the probability of solubility at that stage. Fixed the solubility problem by almost 100-fold, 50-fold, but then had some problem with PIC50. And then finally, after third iteration, everything is green. And compared to AZ0, which is a compound that was what we were interested in learning from. And so as you can see, if you were to develop a compound, we would continue to modify this compound in some fashion, and it takes a lot of work, and it's very difficult to get to something like this very, very quickly. And that's exactly what happened. So this was a kinase compound. As you can see in the beginning, there was a lot of, if you're familiar with the field, you know, if you see anything about this bar that you see here, then that's bad, because that means that this compound is inhibiting a lot of different kinases. And by the time this is all finished, they're only hitting just a few kinases, which is good because they're not off targets, or then the risk of off target is actually reduced. All right, so then coming to drug safety. So what I talked to you before, the drug design phase, this is where most people are interested in, particularly this is where most of the data is available in public space. Drug safety data is not really available so much. It's usually within companies that do this, because you only need to go to drug safety if you actually have getting close to a compound that's going to become a drug. So what happens in drug safety? As you know, drug safety makes a lot of noise in terms of the news, right? If a drug in clinical trial kills somebody or injures somebody, that's going to make a lot of negative news for the company, and one needs, and also for the drug itself. Even if it's a wonderful drug, you know, you've seen this in the COVID phase. You know, there are drugs that had to pause the clinical trial, although sometimes it couldn't be attributed to the drug itself, because it's a blind testing that you're doing, and a review board needs to come to it to figure out what actually happened, because random things happens during a large clinical trial. So the drug safety is something which is very dear to AstraZeneca. We put a lot of effort into it and try to reduce it upfront. So one of the things that happens behind the scenes is before it goes into clinical trial, any drug goes into clinical trial, phase 1, phase 2, just talk of phase 1, we do a lot of preclinical work. And not just because FDA requires it, but also because we want to understand things about the drug, right? So one of the things that we do is we test this in, any company like AstraZeneca size would test in thousands of animals per year, because there are many drugs that are going into the pipeline. And one of the things that we do is we look at what's called as a growth plate size. A growth plate is basically where the long bone ends and then where the junction is. So the size of the growth plate is a very good determinant of how much the bone is actually going to grow. So that's a good indicator of the growth, the development disorders a drug might actually cause. So what we normally do is we look at this growth plate, and you can see there's lines that are drawn. These are painfully drawn lines on an image. Normally, this is not done this way. You just have a pathologist looking at it and say, oh, yeah, that growth plate looks good. It's qualitative. But in reality, we want to be able to actually quantify this growth plate for every drug across many different animals. And it's a very hard problem if you're going to sit there and actually do this all the time. So this is where deep learning is a fantastic, something like convolutional neural network is fantastic for this, is to actually automatically quantify the size of the growth plate. So this slide is from Dan Sutton, who led this effort, a really nice piece of work with collaboration with a company called Halo, where we started to look at how does the AI results compare with respect to the pathologist. Pathologist is an expert, right? And we spend a lot of time curating the data to compare the two. As you can see, the red is on the y-axis is the measurement of AI versus what is the manual curation, and on the green is what is the pathologist versus pathologist. How frequently do they agree? So as you can see, the AI actually has a much higher correlation score, R-square, than the pathologist to pathologist agreement. All right, so we only have 2 minutes left. So I'm going to very briefly go through this. There's also other opportunities such as looking at tumor sizes, etc. You can look at, pathologists need to look at what tumors do we have? Is it drug-causing tumor? How the drug working, etc., etc. So here's another example where a pathologist needs about 35 hours per study to actually come up with the same result. An AI algorithm would do it in 1 hour. So then comes to the question, can AI help reducing animal testing in pharma? So there's many different places where this can be applied. I'll give you one quick example and follow with another one that's very close to my heart. So here is, the previous talk, we talked about a compound having good PK parameters. And PK parameters, there are many to measure. It can have bioavailability, clearance, volume distribution, et cetera, et cetera. So we try to predict, can we predict PK from compound structure and other data that we actually measure? So this was a really good example of simply taking something that's already built in externally, particularly from Regina Barcila's group, and fine-tuning that. And Daisy is part of this collaboration with MPDS at MIT, and fine-tuning that to what we need internally. And what we find is that it was very helpful for us to use what Regina had developed to come up with an accuracy for most compounds within less than twofold in terms of where the actual parameter actually lies. And that's enough in many cases, or most cases, in the early stages of the design to say this compound is good or not. But that's not enough to eliminate rat or animal research. Many things need to come to that, and that's something that we're working very, very strongly on. And my next example actually goes towards that much more closer. Something that we are really interested in is, can we predict the toxicity in clinic early on with the drug. So the idea is we know that gene expression and the great technologies, NGS and single cell sequencing, et cetera, that's going on, that are out there, but can we use that in a meaningful way to predict toxicity in before the drug goes to clinic, because we spend a lot of money otherwise. And animals, and work with people and testing it in humans. So this is a collaboration with Karolinska and the spin-out of Karolinska called Predictomics. What they did is on this gene expression space that we've tried to fine-tune for DILI. So DILI is drug-induced liver injury. So if you take Tylenol, you know that Tylenol and alcohol doesn't mix, right? I mean, it's one of the major warnings, and if you don't try to risk it, do not take Tylenol and alcohol. It's a major issue. It will cause DILI for most people in high quantities. So what we want, one of the key reasons why drugs fail is because of DILI in clinic. And also drugs fail because of this post-marketing, because people didn't really test this extensively enough. So what we did was we collaborated with them, and they had come up with this idea about combining the Broad Institute cMap data with the NCI60 GI50 data, which is cytotoxic data, coming up with a set of compounds that are known to be cytotoxic, but had many different gene expression data in the cMap data. But not just that, there's something that's smart that was done, was they used latent distillate allocation, or LDA, or topic modeling for people who are not familiar with this. The idea is that imagine you had patient 1, patient 2, patient 3, and you monitor what they are eating, so juice, candy, salad, nuts, etc. And you can come up with something like this that says patient 1 has a signature relating to juice and candy, that's 95%, salad and nuts, maybe 5%, while patient 3 is, or patient 2 is more on the salad and nuts phase versus just sugary stuff. So that's LDA in a nutshell. They used that LDA to come with combining gene sets that are correlated with each compound to come up with a toxicity related gene set that is more relevant. And they use that and to apply to our compounds, where we were able to test it across. This is one test that shows 50 compounds, but we tested for many others, and we compared the accuracy with respect to other assays that we had. As you can see, gene expression based assays are very comparable, or sometimes even better than other assays that we had. One of the key interesting thing of this was, as an example, was one compound that went into clinic. It induced DILI within few minutes in some patients, and the drug was withdrawn from phase 1. And this is a unbelievable disappointment for AstraZeneca. But this particular compound, according to gene expression, was one of the top 2 worst compounds in the according to the training set it had. But it never saw this compound before. This is all about blind prediction, and it informed that this is really, really bad. So in retrospect, this would have been a great impact for us. If you knew this before, you would have done something about this. So I will wrap up with one final thing to say industry academic collaboration is extremely important, because we both bring different things to the table. Here's an example of using something just from the academy and trying to apply into in the industry setup and completely failing at it. So before that, I had showed you all the success. So here's a failure example. And so there was a really high profile paper in Cell Chemical Biology that claimed that they're really good at predicting drug toxicity. So we brought this internally. This is the accuracy that the paper has shown in black. We brought it internally. We rerun the simulations. We got similar results, which is in this thin black. And so we were excited. So we can reproduce it, great. Then we tested it on our internal compounds. As I said, this is our benchmark. This is what it's supposed to be. As you can see, the ROC curve is basically close to random for a different classification of DILI. So mild and moderate is basically random, even worse than random in some cases. And for the severe rate, which is more easy to predict, it does have some value, but this is not something we're going to be using. So this is an opportunity for, to rethink about, how it is going to be utilized in industry is going to be very different from how it is going to be utilized in public data sets. So with that, I'll just begin to wrap up and say the three big opportunities that I see are patient stratification. Example is, if peanuts were to go to clinical trial today, and if you didn't know that that drug was a peanut, that drug, if it was for diabetes, will fail because it'll have bad rashes. Some people will die, and you don't know anything about this drug. The drug will fail. So the question is, can we make things like peanuts succeed? And this is happening with a lot of drugs that are completely safe in many ways, but the genetics of the patient determines it, and we don't know anything about it, and the drug fails in the clinic. And that slows us down. Drug combinations and interactions is another. Combinations, I think Regina talked about, others talked about it, as Marinka talked about, the identifying both the combinations into the treatment as well as what drug is my drug going to interact with. And finally, drug affinity, which is still a very difficult problem to solve. Can we identify, can we get better at using AI to predict affinity, both for targets as well as off targets? With that, I'll stop, and I'll acknowledge many people who contributed to this, and thank you for the organizers.
Thank you, Bino, for sharing these insights with us. It was really great to hear about opportunities and the challenges of modeling safety and toxicity early on in drug development.